Awesome, yeah. So company's Phenomic AI. We got started in sort of mid 2017. I joined early 2018, and the work was really based on that of my co-founder, a guy called Oren Kraus, who was at U of T, and then working with sort of Brendan Fry's group and some of the guys out of the group that now has become sort of the Vector Institute there. So as a company, we got going initially more of a software focus and looking at how we could develop tools for pharmaceutical research. Some specific problems here. Although realized pretty early on, perhaps the best application of the tools we're developing, the way we could move the fastest was to not think about, how we can assist pharma companies themselves, but in fact, as a company, as Phenomic, start to develop our own targets and our own drugs. So this is the story to date, and this is where the funding that we've sort of brought on board now is focused on, but how we can really start developing our own medicines using the machine learning and AI tools that we've developed. So I'm going to kick off with framing the problem. And unlike everyone else today, I'm going to be starting with the biology that we're looking at. Well, I mean, we have a pure biology focus. So I'm going to be talking about the biology to start with, and then digging into the tools that we're using to sort of solve this biology, and then looking at,
sort of how we've applied this in the drugs we're pushing through. So on the biology side, the specific problem that we're looking at is in cancer, and specifically solid tumors. So traditionally, sort of in history, I don't know if you can see my pointer here, but the first wave of drugs that came through in sort of the early 1900s were the mid 1900s were the chemotherapies. And so these drugs are pretty unpleasant and toxic, and they just sort of kill off every single cell in your entire body anything that's growing in your body pretty effectively. They are still the best weapon we have against cancer. So more recently, people said, how can we more specifically target the cancer cells? People develop these therapies called oncogene-targeted therapies, and they look at specific genes in cancer that are upregulated. These drugs have been very effective in certain cancers and typically extend lifespan, but quite quickly, because it's a single gene that's being targeted, the cancer will mutate this gene that's being targeted, evolve to overcome this therapy, develop resistance, and the disease progresses. And as a result, 70% of tumors really aren't treatable with current therapies. Most recently in blood cancer, there's been huge success with a type of therapy called immune checkpoint inhibitors. And here we basically look to reboot the immune system. The immune system is sort of already quite programmed to hit cancer. But what cancer does is it shuts it down through a number of pathways. The immune checkpoint inhibitors, they reboot the immune system, which can come along and then kill off the cancer. These are very effective in blood cancers and 1 or 2 solid tumors. But the vast majority, especially sort of colorectal cancer and other diseases like this, they're just not effective. And so as a company, our goal is to think, how can we identify new drugs, new targets that are going to improve on these immune checkpoint inhibitors and then get them into patients where, we can make a real difference. So more specifically, how do we frame this problem? So if you look, here's 3 different tumors, or images of 3 different tumors. On the furthest left-hand side, we have a tumor that's likely to be responsive to an immune checkpoint inhibitor. Here, the immune system's already quite active in the cancer. These immune cells are killing the cells and the cancer cells, and they just need a boost. On the further side, we've got cancers where really it's looking pretty poor, and there's no immune cells whatsoever. But the vast majority of patients with solid tumors present with what we have in the middle here, which is a cancer where the immune cells, are around the place, but they're just not able to quite get into the tumor and kill stuff off. And this is just basically a case where the cancer has shut down the immune system in a way that it can't kill the cancer cells. And so the moment everyone in the pharmaceutical industry focused on this problem, and this is pretty much sort of the holy grail of cancer at the moment, is looking to directly target the interaction, how cancer cells themselves switch off the immune system. But more recent studies in sort of academia have shown that a lot of the shutting down of the immune system isn't occurring directly from cancer cells talking to immune cells, but actually occurring through other key cell types in the environment that sort of indirectly then turn off the immune system. This is a more complex problem because we've not just got 2 cells talking to each other, but we've got 3 or more cells creating an overall state of immune suppression. And so our goal as a company is basically trying to say, how can we find new therapies, new drugs, which aren't directly targeting the immune cells or cancer cells, but other cell types in the environment in a way that's going to let us switch the immune system back on. And the challenge that, why this is sort of a perfect challenge for machine learning and why we think we can have, make a real dent here is that today's pipelines just aren't designed for these more complex sort of systems with multiple different cells. So if you look at how pharmaceutical industry works today, typically assays involve basically taking sort of the cells that are growing together or little cultures of cells in a dish, blending them all together and sort of mashing them all up, and then taking a measurement across every single cell in this system, sort of an average measurement. And so what you do here is you add a chemical to the system, you sort of run a little chemical reaction, and out pops a little sort of number, just a single dimensional number, which says, is this cell alive or dead, or is this protein that we're looking at up or down? And this leads to a situation where it's really hard to understand, which cells are being hit, effectively. Where is there an off-target effect because we're just killing off the cells when we're actually meaning to be changing a protein level? It's really hard to understand what's going on in these systems. And that's just a feature of the way we're doing experimentation today. So at Phenomic, the way we handle, sort of tackle this problem and where we're applying machine learning is trying to understand, in these systems, what's going on at the level of individual cells. So how are individual cells, when there might be a mixture of 3 or 4 different cells responding to the drugs that we're looking to develop. And so the way we do this is we basically use 2 different data sources, principally imaging data, where we capture photos of these cells illuminated with antibodies, antibodies. We use benchtire ourselves to identify antibodies for these tests. And so we basically illuminate the cells with different fluorescent antibodies. We then capture images of them using automated microscopy that can capture images of, it'll capture thousands to hundreds of thousands of different images of these cells. We then use machine learning, which I'm about to dive into, to process this imaging data with the goal of identifying, which cells are responding to drugs, which ones aren't, and which ones aren't, and deconvoluting, the full set of effects that drugs can be having on these cells. More recently, we're now including genetic analysis as well. So we sequence the genomes of these cells, and we can then use the genomes of these individual cell types, that we relate also to the imaging data, but then we can use these genomes to map them back to human tissue data to say, do the cells we're creating look like disease cells in humans? Do the drugs look like they're having an effect, that we want in humans as well? So this is sort of our origins on the tech side, and this is sort of the machine learning. We're basically using 3 different machine learning tools for analyzing the data we're generating. And so the first tool is called multiple instance learning. The second is called weakly supervised embedding. Both of these are deep learning tools. We also use traditional image analysis to help us with interpretation. So the first tool was developed by my co-founder, Oren Kraus, and this is called multiple instance learning. Basically, it's deep convolutional neural network with a specific pooling layer, or sort of, that performs global average pooling over sort of the layer before it, which basically, and you basically use a noisy AND function at this layer, and this leads to this sort of concept of multiple instance learning. We're looking for specific features in an image or specific regions of an image which correspond to there being, it having a positive phenotype or sort of positive example in that image. We demonstrated pretty robustly that multiple instance learning outperforms, traditional feature approaches here, both in terms of, but basically, and here we've not got the traditional ROC AUC machine learning measures. The pharmaceutical industry works on these Z primes, is what people understand. So we actually look at this measure Z prime, which measures the separation between sort of positive and negative controls, sort of more of a t-test type score. And we show quite concretely that the MIL, is more effective at identifying differences between sort of, positive cases and negative cases than the traditional approach. We can then take this sort of multiple instance learning and apply it to screens of different targets and drugs that we perform. So we take negative controls where we, the phenotype we're not interested in, or sort of the cell, the healthy cell type as such. Sorry, the disease cell type as such, and positive controls where we've done something on purpose to make the cell look more healthy, or we've just taken healthy cells. And then we screen a number of targets and drugs, take images of them, process them with MIL, and look to determine, which of these images looks more like the healthy controls versus the disease cells, indicating we found a target or drug that's having a positive effect. We can then sort of, it's quite important, especially for talking to investors and biologists, to be able to show which cells have the positive or negative phenotype, and whether the drug is actually making, these images look more like the positive or negative controls. So we can backtrack, we can sort of do saliency mapping on this and identify, which phenotypes, basically provide, an output to the biologist to say, for double checking, this actually looks like the positive control and the drug we care about. So weakly supervised embedding is the second tool we use. And this was developed, I did a bit of stuff myself around my PhD on this, looking at traditional features. Juan Cecido at the Broad Institute developed this through deep learning more recently. And so in this case, we're basically looking to embed single cells, images of single cells into a space which lets us understand, which drugs or treatments have had an effect on cells versus which ones haven't. So the way we do this is that, in every screen of a drug we perform, we've got metadata which says, which conditions are different to each other, which conditions are the same. And we also know that all of the cells within a single condition belong to that condition. And so we basically present this sort of classification task where we say, we basically use a neural network to identify a single cell object. We pass that to a second sort of embedding network, which is we basically train to say, which condition does this cell belong to? So here, there's an example where we've got a probe, SMAD4, which is in the nucleus of the cell here in this image, and it's in the cytoplasm here. So there's a set of conditions here. I don't know if you guys can see this, but in condition 2, you see that in every single cell, this probe SMAD4 is in the nucleus. In every other condition, this probe is in the cytoplasm. In contrast here, this probe, this cell, this probe SMAD4 is in the cytoplasm. In all these conditions, it's in the cytoplasm, it's not in the nucleus condition. It's not in the cytoplasm in condition 2. By then taking, one of the later layers of the network that's trying to classify, which condition this cell belongs to, we can pull out this sort of embedding or sort of barcode, which shows, quite nicely that you get a strong signal for condition 2 and a weak signal for the other conditions. You then get the inverse for the one where it's in the cytoplasm. And projecting this down onto two-dimensional space, we see that this condition TGF-beta, so I've revealed the conditions here now, this condition TGF-beta, which we know, induces SMAD4 to go into the nucleus, looks different, or, stands out from all the other conditions. And so applying this at scale then allows us to say, is a treatment having an effect on the cells or is it not? Which is really valuable when you're doing massive screens of tons of different conditions and tons of different probes and readouts. The final thing we do is we use, a number of segmentation algorithms, some deep learning, some more basic, to then, extract, identify regions which correspond to different cells in these more complex images. We can extract features telling us what's going on at the single cell level, map these back onto the weakly supervised or the MIL sort of plots, and this helps us understand, what's going on, in these images as biologists. Everything's now integrated. What we've done is worked pretty hard, the software team has worked pretty hard to integrate this on AWS. So we now use a set of, we use a standard sort of Kubernetes, Docker sort of containers to store everything, so we can spin stuff up on the fly. Everything's held in, sort of S3 and stuff. And the guys have most recently upgraded to TensorFlow 2. So all in all, we can now analyze these images quite nicely on the fly. And we use a couple of key sort of open source platforms specific for microscopy. Omero is one of them for managing and storing the data and allowing biologists to interpret what's going on. I'm not super versed on this, so I can't answer too many questions on our stack at the end. So jumping into how we're using this quite directly to solve this problem of identifying new drugs to target cancer with. So the workflow we've developed is we basically identify new drug targets. And so these are sort of, things sort of fundamental units or proteins we think are driving the disease. We identify these targets from omics data. So this can be genetics data, proteomics data, or sort of hits from whole genome CRISPR screens. Basically, we get very long, maybe hundreds to 1,000 long lists of targets we think might be driving the disease. We run a forward test in which we take the protein itself, we add it to different cell types that, in a healthy state and say, does adding this protein to the healthy cell make it look more disease? In the reverse test, we take one of these more complex models with tons of different cell types interacting with each other, and we take a drug that inhibits the target, add it to this sort of complex model, and then use our machine learning to say, are we making this disease state look more healthy? And then finally, hits from these, once the target's made it the whole way through, we develop our own drugs against the targets, our own therapeutics against the targets, which we then put into animals for further testing.
and that sort of precedes the clinical trials. So I'm going to run through the case, the first sort of proof of concept we did with our seed funding that enabled us to now raise our Series A sort of round. Our pre-seed and now raise a sort of a large seed round. So we started out by taking sort of public genetic data from both the Cancer Genome Atlas and some single-cell data sets we sort of gained access to. We analyzed these using just traditional sort of statistics and bioinformatics approaches, and identified a set of targets that we thought were highly upregulated in solid tumors, correlated to cells that we know are hypothesized to be immunosuppressive, and pulled out a list of a couple of hundred targets we cared about. We then developed a set of sort of experimental models that would enable us to test whether these targets are creating these more immune-suppressive cell types. So a set of different assays we created. Some involved purely immunofluorescence. Others looked at how the cells are behaving in sort of different environments. And a final set of assays looked at, whether cells are becoming more invasive and driving, sort of these invasive phenotypes into neighboring tissue. We profiled all of these targets. This is this is one of the smaller matrices. We did much bigger experiments than this. We can see here, quite nicely, a range of different effects targets are having against different markers of, what sort of state the cell is in. One target here is target K, was very strong against against this set of different markers in a way that we thought was interesting. So we sort of dug in and showed that, quite nicely, it's having an effect on this cell type, or making a certain cell more immune suppressive. This matrix here, so every single data point here is a set of different, is about 10 or 20 different images from, 2 or 3 different experimental replicates. What we've done is we basically, across every single different probe, we embedded all of the single cells from all of these different images. We then took, the mean sort of embedding of a bunch of these individual cells, compared it to the mean embedding for every single cell in every single condition for this probe, and said, how different is an individual condition compared to the average or the mean of everything. And this we found was a very effective way of identifying, which conditions is a drug or target having an effect in versus which ones isn't it. Here we're looking at this multiple instance learning again. So we're saying, in this case, we just performed a dose response. We're saying we know the drug or this target's having an effect. So we use the positive control as the highest dose and the negative control as the lowest dose. And what we're looking for is to say, see a nice dose response curve or dose dependent effect, which gives us really good confidence that this target is really hitting these cells effectively, having an impact on these cells and not just an artifact. The second thing, and this is really what pharma struggles with today, is analyzing these more complex models and saying, if we block the target using a tool inhibitor, so just sort of a reagent that inhibits this target, do we see a nice effect? And these images are way too complex, especially way too complex and way too variable for today's, normal feature-based approaches. And so here, deep learning is critical. And so for one target that we identified, we basically, we tested a number of inhibitors of, maybe 5 or 10 different targets in these more complex models. And we found an inhibitor of one target that was highly effective at basically blocking, block the proliferation and sort of, a number of other features around a specific immunosuppressive cell type. Again, applying MIL, across a number of different doses, we saw a nice dose-dependent response. So this has given us, in total, a lot of confidence that this target is driving immune suppression in a number of different tumors. Through all this conviction, we've now raised significant capital to develop a set of therapeutic antibodies. So sort of read into this if you're interested. So a set of therapeutic antibodies against this target, and now basically, as of I think now last week, we've got some successful hits against this. So over the next sort of couple of months, we'll be shifting into in vivo or mouse models to determine whether they work there as well. And if we've got conviction here, sort of over the next sort of 2 to 3 years, we'll then start building the case and the strategy for moving into clinical trials with the ultimate hope, that we can get this approved and into patients. So for that, I'd like to say thanks for listening, everyone. And we've got a great team, mixture of both sort of bioinformatics, machine learning, and wet lab biology. And we're growing all the time. And we've also got advisors. So Oren, my co-founder, developed a lot of this technology early on.
Jimmy Barr was a key early advisor on the machine learning side at the Vector Institute. And then Boris and Brendan at the moment are guiding us on the biology side, with more to join soon. Early on, we came out of the University of Toronto. We've had some funding from Mars Innovation, and a lot of this technology is associated with the Vector Institute. And then our investors, we've got a range of different investors, both here in Toronto, over in San Francisco, as well as the UK and as far abroad as Hong Kong too. So with that, I'm going to stop sharing the screen and open to questions.
Yeah, awesome. Thanks so much, Sam. Super insightful and interesting. It was kind of insightful to see how each of the different businesses kind of addressing this drug discovery kind of biotech industry as a whole. Yes, definitely claps for Sam. Thanks so much, man. It's strange kind of it's having it all online and not seeing an audience and everything. But yeah, good turnout. And I'm sure everybody's really appreciative of that. So we've got a question now here from Kenneth. Kenneth says, "Hi, Sam. That was a fantastic presentation. Solid tumors are so diverse in biology and therapeutic response. How do you account for this diversity when building your models at a single cell level?"
That is a fantastic question, Kenneth. Yeah, so tumors are super diverse. So everything we do is this might be not. I might not be answering your question quite first time. I'll give two answers depending on how you interpret your question. So if you're talking about models in terms of mathematical models, so we're not doing that. We're doing phenotypic discovery. So we actually take wet lab biological experimental models and test stuff on them. And so, if we test them, and so, we're not having to predict anything here. It's just purely, does it work or not? If you're talking about experimental models, then we use cell types from a number of different cancers and a number of different patients, and test our sort of targets and drugs across maybe 20 or 30 different models from different people, individuals who've had cancer, and we're looking for hits to be effective in a significant number of these sort of models. So we think that a subset of patients will respond.